common metaphor for the brain is a computer. Central to the design, the production, and the functioning, of course, of a computer is its wiring diagram. A computer consists of many components which are connected with electrical cables, wires, through which the signals propagate. And to make the production and repair of computers easier, often these wires are color-coded. The brain consists of billions and billions of components, neurons, nerve cells, and also connected with wires. These wires are actually part of the neurons themselves. We call them dendrites when the electrical signal is propagated towards the body of the neuron, and we call them axons when the electrical signal is propagated away from the body of the neuron. Central to a challenge to understanding how the brain functions, how it works, is to decipher, to describe the wiring diagram in great detail. What neurons are connected to what trajectory, what route do these wires take, and equally importantly, how does this all come about during development? With the technologies that we have today, even the little humble mouse brain is far too complex to study in its entirety. A complete description of the wiring diagram of a mammalian brain, let alone the human brain, is not yet on the horizon, certainly not by 2020. Instead, what scientists do is to focus on bits and parts of the brain. We call them models or model systems, and we hope, of course, that our findings, our conclusions from these models or model systems are generalizable to the rest of the brain. In my 18 minutes of fame today, I will share with you an exciting genetic approach we've been taking for a while to study one bit of the mouse brain, the olfactory system. I will explain to you that it develops largely in a self-organizing way, ex nihilo, if you wish. We have developed and applying genetic tools that literally allow us to stain wires, axons and dendrites of neurons, so that they appear color-coded. Why we do this in the mouse? First, it has a very well-developed olfactory system, and secondly, genetic manipulation of mice is easy or relatively easily. That's our specialty. Our team has been using for 20 years now the methods of gene targeting technology. We've been established 25 years ago. It's a complex, time-consuming, and somewhat tedious process, resulting in strains of mice after culture of embryonic stem cells, DNA engineering, and microinjection technology. And the details, of course, I will spare you. This is the first mouse that I engineered back as a junior scientist in the lab of Richard Axel, Columbia University, New York, 20 years ago. And I engineered the mouse strain such that all the neurons, olfactory sensory neurons, express a particular bacterial enzyme. And this enzyme converts a chemical that has no color into a blue precipitate. So wherever there is a cell and an axon expressing the bacterial enzyme, there will be a blue precipitate. Olfactory sensory neurons are the detectors. These are the neurons in the nasal cavity of a mouse or in human that directly detect the smelly substances in the inhaled air. In the nasal cavity of either mouse or human, olfactory sensory neurons cover the back end and top end. They appear as a homogeneous blue sheet here because there are millions of neurons you look at the same time. Each of these neurons has one axon, one wire. These axons penetrate through a perforated part of the skull and then will terminate in the cranial cavity inside the skull in a structure called the olfactory bulb. So that's the overall anatomy. By the way, this little organ here at the bottom has attracted a great deal of attention. It has the shape of a hollow cylinder, and the inner lining consists of hundreds of thousands of specialized sensory neurons called vomeronasal sensory neurons. What these neurons do is to detect pheromones, molecules with which animals communicate. Essentially, it deals with sex and violence, hence the great deal of attention it's been getting from scientists. But I've got news for you. In humans and in other great apes, there is no longer a vomeronasal organ. So now what happens to the axons, the wires of these olfactory neurons, once they get into the cranial cavity? They terminate in the olfactory bulb in little structures called glomeruli, of which I highlight 3. The recent count is that there are about 3,600 glomeruli in the mouse olfactory bulb. On the left side is the axons of the vomeronasal sensory neurons, which terminate in a special part of the olfactory bulb. These glomerulus is a Latin word for a knot, like a ball of wool, They were discovered in the 19th century, so in the 1800s, and it was always clear from the beginning that it represents some kind of iterated unit, some element or component that is repeated, in this case 3,600 times. But what exactly does it represent? Well, it took our genetic tricks and tools to visualize this very, very readily. So how do we go about now? Just color coding, just visualizing those olfactory neurons that express one particular odorant receptor gene. What I didn't tell you yet is that olfaction is mediated by the largest gene family in the genome. A mouse has 25,000 genes, and no fewer than 1,200 genes are odorant receptor genes, genes encoding odorant receptor proteins that detect the smelly substances. In human, there are about 400 olfactory receptor genes. So I came up with a trick in which the expression of a particular odorant receptor gene is coupled to that of a color code. It could be that bacterial enzyme that I referred to a moment ago that gives that blue precipitate. And in this particular example, it's a green fluorescent protein, GFP. It comes from a jellyfish, and when we shine blue light on it, it shines back green light. So in this mouse strain, the few thousand neurons that express one particular odorant receptor gene, one of these 1,200 genes, they will become green fluorescent, and only them. So you get the point. We can color code, visualize just specific wires. Here is one example. The odorant receptor M71 has several ligands known, several molecules that interact with it, and one of them is benzaldehyde. It has a sweet almondy smell, just like the Italian liquor amaretto. That is essentially benzaldehyde. There's a few thousand of these neurons in the nasal cavity of a mouse at the back and top end. Each of these neurons has one axon. The axons go through the perforated part of the skull. They take a very broad trajectory, and they all come together almost magically into a single dot, in a single glomerulus, just one on this side of the bulb. And this is a mouse that's about 3 weeks old. It happens very fast after birth. One of my all-time favorite pictures, this is the glomerulus you were just looking at, on the right bulb, what we call the medial side, and on the other side of the same bulb, there is another glomerulus, and then the whole thing is duplicated in the left bulb. So there are 4 glomeruli per mouse that represent information that is received by this odorant receptor M71. You can see from this picture that these positions are not random. They're conserved. They're not exactly the same, they're not stereotyped, but they're highly conserved. By the way, we don't really know if the particular position has any information to it. It's just what we see in such mice. We can ask then the next question. Given that the positions of these glomeruli receiving information of neurons expressing one receptor is conserved, is also the trajectory, the terminal part of the route the axons take to form a glomerulus, is that also conserved? For this, we turn to another strain of mouse. We have many, many strains in the lab in which we genetically manipulated a receptor gene that's similar to M71 called M72, and we manipulate it in such a way that when these neurons express that receptor, they also express the green fluorescent protein from jellyfish. So here are the 4 glomeruli at conserved positions. We can now use this mouse to image glomeruli and make what we call 3-dimensional reconstructions using a special microscope called a 2-photon microscope. And a 2-photon microscope can look much deeper into opaque tissue, which is the olfactory bulb. The middle of this image is the glomerulus itself. The diameter is the long diameter about 80 micron, the narrow one about 30 micron. There is one large bundle of axons ending up in it. There are several smaller bundles of axons and very fine ones. But importantly, all the axons that are color-coded green all end up in this glomerulus. Amazing example, I think, of biological specificity. Since we have a strain now, a mouse strain, we can just breed many more mice and look at many more mice and ask, is this particular way a glomerulus is formed, is that also conserved from mouse to mouse? The answer is no. The axonal plexus, so the terminal end of these axons, this is the one I just showed you in the previous slide. In the other bulb of the same mouse, you find 4 large bundles of axons, all coming together and make a glomerulus. Then in the mouse from the same litter, we call that littermate, we have many smaller bundles of axons, and interestingly, the top right, one bundle that seems to shoot too far and loop back. And then finally, this is one of the more complex structures, many fine cables coming together, crisscrossing, going apart again, and then ending up in this one glomerulus. And that's what matters, of course, in the end, that they all come together in a single glomerulus. So when we look at these pictures and with other results, what we see here is that this system is self-organizing. We imagine that axons of neurons expressing a given receptor are looking out for other axons of the same type, that have some kind of chemical affinity with each other. And when they find each other, and there's enough of them, then they coalesce and form a glomerulus. It's self-organizing. Now, that's an hypothesis, that's a model. It requires a lot more work, as we scientists like to say. And what we particularly need is actually movies here, or movie clips. The metaphor I can use is that of trying to deduce the rules of a football game by looking at a few still pictures. You'll never figure this out. But if you have a movie or movie clips, then you can deduce at least part of the rules of football. With cricket, that's hopeless, by the way. Now, there's one thing more. We have now shown that all the neurons that express a particular odorant receptor send their axons in such a way that they coalesce into a few glomeruli at conserved positions. I've shown in other experiments, by manipulating the receptor itself, that it's really the odorant receptor that determines these positions, which is perhaps also the most logical. But even more so, we can replace the odorant receptor with another receptor that's not an odorant receptor, the beta-2-anergic receptor. Some of you may know about it because it's used to make drugs against high blood pressure. And the neurons that we have now artificially induced to express that receptor, instead of the odorant receptor M71, they somehow manage to coalesce at conserved positions in the bulb. Pretty amazing, right? These neurons have never expressed this. In normal mouse, they don't do. But yet, when we create this artificial situation by genetic manipulation, there seems to be space in the bulb to accommodate a novel glomerulus that didn't exist before. So I dare to speculate here that this fantastic biological specificity of wiring occurs ex nihilo, out of nothing. There is no template, there is no program, there is no grid. There's nothing in the olfactory bulb that attracts all these axons and lays them out in thousands of glomeruli in 1,000 points of light. It is the axons of the neurons themselves that are doing this in a self-organizing way. So the wiring diagram is such that axons of neurons expressing a given receptor dynamically form these structures, these glomeruli, and without, let's say, the green-coded neurons, there would simply be no glomerulus, there would be nothing. And here, the metaphor of the computer falls apart, breaks, as every metaphor in the end does. It's not the case that the olfactory bulb, and perhaps the brain in general, has some pre-configured slots and positions in which wires of direct color code just have to be plugged. No, instead, it is, at least in this case, self-organizing. Now, that has a lot of advantages, this flexibility, this plasticity. If you think about it, particularly in an evolutionary sense, if a new odorant receptor gene occurs during evolution, new smelly substances can be detected, and a new glomerulus will be formed somewhere in the bulb at conserved positions, and it can be integrated nicely into the system. So this self-organization is a powerful engineering feature. 1,200 odorant receptor genes in the mouse genome, scattered everywhere over many chromosomes. So what we need to understand ultimately is what are the mechanisms that allow an olfactory neuron to express just one of these genes. That's quite unusual in biology, that of a large gene family, cells express just one of them, and that's pretty well accepted. For a long time in our field, we thought it may happen by a process of gene rearrangements. Now, what is that? The one receptor, one neuron rule smells very much of lymphocytes. These are white blood cells. There are two types of them. The B cells make antibodies, and the T cells make T cell receptors. Now, every B cell makes only one antibody, and every T cell only one T cell receptor. There would be more than two. There would be noise in the system, autoimmunity, and what have you. The way by which lymphocytes do that is by irreversibly changing their own DNA, irreversible genetic alterations. This is very well described in the 70s and the 80s. So in our field, when these odorant receptor genes were discovered in 1991, there was for many years the idea that they would also undergo some irreversible genetic changes, so that a neuron chooses a receptor, it's locked in, and for the rest of its lifespan, it can only express that receptor. Right? That's an attractive idea. To show that, we took a rather extreme approach, and we cloned mice from the DNA of neurons expressing a particular receptor, our favorite receptor, M71. You know about Dolly, the cloned sheep, reported in 1997, and a year later, a team at the University of Hawaii in Honolulu developed a cloning technique for mice. I heard about it, contacted them. I made many trips to Honolulu to learn this technology, brought it back to my lab, and this is the guy who developed it. He's also a good friend of mine, Teru Wakayama. In his left hand, the black mouse that he holds is probably the most photographed mouse in history. It's the first cloned mouse, Kumulina, and she lived a very good life there in Honolulu for 2.5 years. That's the average life of a laboratory mouse. So Teru gave me a bunch of slides, very nice slides about the process of nuclear transfer that I'm going to show you now briefly. Cloning by nuclear transfer of animals happens under microscope with microinjection technology. A key aspect of this technique, by the way, is a special piezoelectric device that came not coincidentally from Japan. The first step of the cloning process is actually the hardest one, which is you have to remove first the DNA of the oocyte, of the egg, before you can add new DNA in. All of us started out like that, just like a single oocyte, whether we are a human or a mouse or an elephant. It's about the same size. We're holding the mouse oocyte with a blunt pipette, a holding pipette, and then with a sharp pipette, we come in and punch a hole and aspirate the chromosomes, which we next expel. So what's left behind is an empty egg, an egg that doesn't have its own chromosomes. It's enucleated, as we call it. And an egg can survive a few hours like that in an incubator at 37 Celsius. And then in the second step, we isolate the nuclei of the so-called donor cells. We call them donor cells because in this experimental paradigm, they donate their nucleus, their DNA, to an oocyte. Very often in cloning, cumulus cells are being used. They are easy to get from mice. And that's why Kumulina has that name. And the trick that Teru developed is to use a pipette that's too narrow. So when you aspirate a cumulus cell in a pipette that's slightly too narrow, the cell membrane ruptures, the cell breaks. But the nucleus, however, is stronger, doesn't break, and in fact, you can squeeze it into a tiny little sausage, as you can see 2 of them here. Then the third and final step, you have now isolated in a physical way with a pipette that's simply too small. You go back and inject in these enucleated oocytes 1 nucleus at a time. That requires a great deal of skill, by the way. It's easy to show these pictures. To puncture a tiny hole very fast. This process is very inefficient. It was very inefficient in 1998. It's still inefficient. About 1 in 10% of the time you get a cloned animal that's born alive. In our case, it was even more difficult because unlike cumulus cells, olfactory neurons expressing a particular receptor, let's say M71, are very, very rare. When we prepare cells from samples from the nose, it's 1 in 10,000 to 1 in 20,000 cells that we can use. Nonetheless, we managed after a while to produce several mice, cloned mice. And one of them is Harvey, a cute, lovely mouse. Also very hard to get it to pose for a picture. And after analyzing Harvey and his clones, his brothers, it was immediately clear to us that they're essentially normal mice. The olfactory neurons in these mice were not all producing M71. They were not monoclonal, but they could produce other odorant receptor genes. So if there had been irreversible genetic changes in cells expressing M71, this mouse would have been monoclonal. All the cells would have expressed M71. So we actually refuted our own hypothesis, which is typically not what you do in science, right? And certainly not writing a grant application to disprove an hypothesis. Nonetheless, we and our competing team got it published in Nature because it laid to rest a dominant and exciting idea that was around for 12 years, but without any kind of outcome. Now, the project was also fun for another reason. Harvey was made in New York. At the time, I had my lab at Rockefeller University in New York. And I called this project, which took 6 years, the Sleeper Project, after a movie of another New Yorker, Woody Allen. In this movie, at the end, the great leader is blown up in an attack. They found his nose, and from the nose, he is cloned again. So I thought, this is my chance here. I wrote a letter to Woody Allen. I figured out where he lived, and asked him, can you please come to the lab and look at our sleeper mice? That didn't happen. I got a letter soon thereafter. It's not an April Fool's Day joke here. It's a real letter. Dear Professor Mombaerts, a small typo. Congratulations on cloning your mouse. I don't know if I would be interested in visiting the lab and seeing your mouse. And now here it comes. However, if I can obtain a few cells from Angelina Jolie, perhaps we can tell. Yep, that really made my day, and you can see I'm not funny enough to come up with something like that. On that note, thanks for your attention.